you're always starting with DNA. What happens after that varies. But we're starting with DNA. You can tell because it's double-stranded. So. Okay, and again, I don't care about your sequence, but whatever you have, just make sure the other strand fits. So helicase is unraveling, just like before. And now we will have part of the polymerase come in again, bring in some new nucleotides. So adenine would link up with thymine, but remember, we're now doing RNA. And what comes in is not thymine, it's uracil. So that's one of the differences. RNA polymerase does not recognize thymine. It recognizes uracil. So that's one difference between DNA and polymerase, DNA and RNA polymerase. The DNA polymerase recognizes thymine. RNA polymerase recognizes uracil. So where you expect to see thymine, you get uracil. Next one, thymine. So up here, adenine. Cytosine is here, so guanine. Now I know somebody's going to ask me, so I'll just nip it in the bud. Why is thymine here if we're doing RNA? Remember, this is the DNA strand. The DNA strand can have all the thymine you want. It's up here in the RNA strand that you have to have uracil. So we once again are doing our nucleotide thing, base pairing. And once again, ligase. Yeah, we'll sew them together. So really, other than the uracil, it's the same process as for replication.
So, is when the DNA is being unraveled, is both the RNA polymerase and the DNA polymerase coming in and doing their own job?
The question is, when we're doing the unraveling, are both DNA and RNA polymerase being used? No. DNA polymerase is only used for replication, and we're not doing replication. The DNA polymerase is only used if you're making new DNA, but we're not.
Oh, I was just wondering if they got the job done at one time.
No,
Replication.
no, it's a different process.
So an example of making new DNA would be like, instead of making new cells, would be like cutting yourself and like killing yourself.
Yeah, we're not done. Now, there's one other difference. Because this actually is what we're getting. Now, we're not done yet. But I don't have anything up here. This is the other difference. RNA polymerase only recognizes one strand of DNA. It does not recognize the other one. So the other strand is not being used. The other strand of DNA is not being used. Now, the term is template. The word template means a pattern or a mold of some sort. For example, the first strand is being used as a template to make the second strand. The second strand is the same as the first, except backwards. So that's the base pairing. The second strand is like the mirror image of the first strand. So the first strand is your template. In replication, both strands were used for templates. Because both strands in DNA were being used to make new strands. But for transcription, only one strand is being used as a template. Now maybe another example would be, I still remember as a kid, my mother would make cupcakes sometimes. And she would take out this cupcake pan, or cupcake tin, whatever. And it was just this flat sheet, but in the sheet there were some holes, so some, I don't know what, some holes of some sort. And she would pour the cupcake dough into these holes, and later on, magically, would out come cupcakes. Well, the cupcake tin was the template. Because whatever shape those holes were, the cupcakes came out the same shape. Or if you're making cookies, you had these cookie cutters, so, make the cookie cutters. The cookie cutters would be your template, because your cookies will come out to be the same shape as a cookie cutter. So those are templates. So the template is simply the model or the pattern that you're going to use to make something new. In this case, for transcription, only one strand is used as a template, the other one is not used. For replication, both strands are used as templates.
So what is the,
What's used to make the RNA?
and then after that?
Well, we're not done yet. Hang on. I know you guys are really eager to hear the end of the story, but we're not done yet. Is everybody okay with this concept of template? Okay. So again, for RNA polymerase, it's only recognizing one strand of DNA, but not the other one. I need to erase again. 4. The DNA continues to separate. So what we have here on top is the new RNA strand. Down here is your DNA strand. And over here is the DNA strand that's not being used. Okay? So DNA is here, RNA is here, DNA up here again, not being used. Well, once again, sooner or later, you get to the end of the molecule, the end of the DNA. So the DNA strands separate, they come apart, just like before. So when they come apart, the upper DNA strand just separates, but the lower one with the RNA now separates. It comes off of the DNA. And now the remaining DNA strand to the RNA has now come off. So the remaining DNA strand now has no more RNA.
Okay, so I was looking down when you were pointing to so the new nucleotides are added on, and then, so that whole thing on the bottom, that becomes the new DNA, so the RNA is the strand, the second strand that broke off.
Say that again.
Trying to figure out how to explain this. So, which one is the RNA strand?
The one on top. Because that's the one that you're forming.
It's a new RNA.
This is the new RNA strand that's forming.
Okay.
See, the bottom one, this is your original DNA.
But then at the end, they fully
They
separate.
come apart.
And
So this one on top, to have it kind of in the same position, this one on top is this. And that's your RNA. The one on the bottom is this one.
You have two like single strands of DNA.
Yes.
Okay.
So two single strands of DNA
That's exactly what I.
and one strand of RNA. The final part of all of this, these two strands of DNA, they were connected. The final stage is the RNA polymerase will bring them back together again. So reconnect, you have your original DNA back together again.
And that's just another polymerase.
Well, it's the same RNA polymerase.
No, I understand that, but there's a name helicase that separates the DNA.
Oh, I see, yeah, it's just another part of the polymerase, yeah.
Sorry.
So what you end up with here is the original DNA. You got your DNA back again, and you have your strand of RNA. The DNA can now be reused. You can do it almost infinitely. Not truly infinitely, everything breaks down, but you can reuse it a lot. And meanwhile, the RNA will wander off and do what RNA does. And that's transcription.
So DNA never changed, the only purpose of RNA was to get information from the DNA.
Yes, exactly right. The DNA never changes. The only purpose of the RNA is to get information from the DNA. Keep in mind, the RNA sequence is basically backwards of the original DNA. So it is like trying to read a letter in the mirror. You can do it, but it's hard. But that's essentially what you got there.
So this whole process is
The
transcription.
whole thing is transcription. And the whole purpose of transcription is to make RNA. So similar to replication, but not identical. Again, the two differences are that RNA polymerase will bring in uracil instead of thymine. And second, that the RNA polymerase will only use one strand of DNA instead of the two. Everybody okay? Questions? Okay, not asking if you're excited, I kind of assume you're not, but as long as it's fairly okay. And as always, I don't expect that you actually understand everything right now, but is there any part of this that when you go home, the RNA is actually now going to be called messenger RNA, little m, then capital RNA, messenger RNA.
The new RNA strand.
Yeah, the entire RNA strand is simply messenger RNA. And it's called the messenger because that is the molecule that's going to leave the nucleus and go into the cytoplasm. And again, even though bacteria don't have a nucleus, the same process still holds. The RNA will go from the chromosome to find the ribosome. So what I said is that the messenger RNA is going to leave the nucleus and go into the cytoplasm. And it will go into the cytoplasm specifically to find the ribosome.
Where DNA
Hmm?
will be made. Where the DNA will be made.
The DNA actually will be reused over and over again, but it will stay in the nucleus. Because after all, every one of us, sperm or eggs, we're making new ones all the time. So as we make new sperm or new eggs, we have to do this process all the time. Yeah.
You said messenger RNA goes where to find?
From the nucleus into the cytoplasm.
To find ribosomes.
Okay, any last questions on that before I erase? Well, you're still right, so I'll be quiet. Yeah, because everything's in the cytoplasm. There is no nucleus.
Okay.
So basically, your pathway, I guess, is simply from the chromosome to the ribosome. Remember, all of these processes, we basically got them from bacteria. And we still use them even today. We may have modified a little. Glycolysis, we never did modify. That one we took straight from the bacteria. But, things like this, we modified it a little, but we still basically took it from bacteria. Okay, any other questions before I erase this stuff? now we start to do translation. Remember, translation is the synthesis of proteins. The synthesis of proteins. So the messenger RNA leaves the nucleus. So we're done with the DNA now. I mean, it'll keep doing it over and over again, but it's a repeat of what we just said. So we now are finished with that. So the translation, the messenger RNA leaves the nucleus, goes into the cytoplasm, and finds the ribosome. The ribosome looks like this. It looks like a snowman. And it's got two pieces, as you can tell. And the pieces are made up of protein and another RNA, ribosomal RNA. So again, now little r and then capital RNA. So ribosomal RNA. It's a second type of RNA. It's made the same way as messenger RNA. The base sequence is different, so the message is different. So again, it does something different than messenger RNA. It's also made by transcription, just like what we just talked about. But ribosomal RNA plus protein makes up these two pieces of ribosome. And these two pieces are described as 30S and 50S pieces, 30S and 50S. So the obvious question, what's an S? Well, before I answer, what's going on is that one of the very, very fundamental pieces of technology that all research labs use, and frankly almost everybody, is the basic centrifuge. You spin things around, and depending on how heavy stuff is, it'll sink to different levels, and that's a very easy method of separating and isolating different molecules, different cells, different everything. But the problem is that weight alone is not sufficient. For example, what I have here is an ordinary old piece of paper, and I drop it, and it kind of floats down and stuff. But then, if I crumple it up into a ball, the weight has not changed, it's the same weight, but now it drops a lot differently. In other words, weight alone is not sufficient to explain the properties of how something separates in solution. So not only shape, but also structure and other things, density, and you can go on for a while with that. So this guy, Svedberg, I'm doing it, Svedberg. That's where the S comes from. He was a Swedish scientist, now a dead Swedish scientist, but a Swedish scientist came up with a system of measurement that would take into account both shape and structure and mass and all kinds of things for centrifuging particles. Now you don't need to worry about that too much. I'm just trying to explain what S is, but it's nothing I'm going to ask you on a midterm, so don't worry about that. It's just simply a system of measurement. But to give you an idea of how complicated it gets, very, very quickly, 30S, 50S, the total ribosome is 70S. So right away, the obvious question, what happens? Why is that 80? Again, off the record here, I'm not going to ask this of you, but again, it's reflecting sedimentation and centrifugation and so on. They get, what?
They have it.
Wait. So the way it works is, well, they aren't bonded together, of course, but the way it works is that if you have two separated particles, so they're not bonded to each other, two separated particles, if that were sedimenting against centrifuging, whatever, you would have 8 different places of friction or resistance or something, of interacting with the solution. You'd have 8 different solutions, 8 different places. But if you just take the ribosome together, so the two pieces are bonded together like they're supposed to be, now you only get 4 different places of interaction. That's why it doesn't add up. Because even though the weight would be the same between this and this, but clearly the shape is not the same, and the structure is not the same. Okay? And again, total amount of friction is not the same. So like I say, it gets very complicated very, very quickly. I'm not going to ask that of you, I'm just giving you background knowledge, information. Tell you a little story.